Hello. Today I'll be presenting on an automated genotype database for associating counterfeit drug evidence. So first, let me provide you a little bit of background on counterfeit pharmaceuticals. This is a growing issue. It's killing over 100,000 people each year, and in the past 2 decades, the number of incidents has grown from 200 to 4,400. One pharmaceutical company, Pfizer, tested 250 of their Xanax purchases online and found that 96% were counterfeit. And this is continuing to grow with the annual trade in counterfeit drugs beginning to rival the trade in illicit drugs, 100 billion to 250 billion. How did we become involved with this? So a private company had a collection of samples from known counterfeit drug packages, and they went and had a DNA lab go and generate data from this over an 18-month period, starting in November 2018 through to May 2020. And so what that lab did is they analyzed 133 samples representing 103 package evidence items. Unfortunately, this lab analysis didn't really produce a lot of useful information. And so then this private group came to us and asked us, using our TrueAllele technology, could the TrueAllele database find more information from this data? So at Cybergenetics, we received this data and then processed it. We took all 103 samples. Someone quickly looked at the data and just gave a rough estimate of the number of contributors. And what we found is the data typically contained somewhere between 2 to 5 contributors. And so then we set up processing for those samples, and then our task was to find connections between different collections of these counterfeit drugs. And a part of this is that we were looking at the throughput of the system. We first received the data in May 27, 2020, and a week later on June 3rd, 2020, we produced the first preliminary results. So what do those preliminary results look like? What I have here is a graphic to illustrate the results, and this is going to grow as we add more results. So initially, in the top, you'll see that we have a circle representing one sample, which is vial and label edges collected by certain lab tech AS in February of 2019. Off to the left, I just have a generic drug picture, doesn't necessarily have anything to do with this study. And then also, you'll see that there's a list below that of manufacturer case, and each case is a different collection of a counterfeit drug. So for this first item, the previous analysis listed it as not a searchable mixture, probably due to something with the complexity. TrueAllele, however, was able to tell us that it expected if we found a matching contributor that match statistic would be a nonillion, which would be 10 to the 30th, so 1 followed by 30 zeros. Now I've added a second piece of data. So this is a metal seal, vial labels, flip cap, and bottom rim collected by a different lab tech, JD, and that's important because different lab techs mean that this isn't just like the lab tech contaminating a sample. And it was also collected in May 2019. And you'll notice off on the left, now we have another case 8. So this was from a different collection. What the initial analysis showed from the previous lab was that it was again listed as not a searchable mixture. However, TrueAllele found a match statistic between these two items of 1 nonillion. So it's comparing these two samples to each other and finding a common contributor between them. Further, there's now a third sample. So you'll notice that this sample is a carton vial patient insert connected by a third lab tech, PN, in January 2020. This is from a different collection, case 19. And what we're seeing is all 3 of these items are matching to each other. Initially, the previous analysis found that these were all not searchable mixtures. However, TrueAllele has now found a cluster of matches. The first 2 items, as we said, matched at 1 nonillion, and then with the third item added, we see match statistics of 10 octillion and 100 sextillion. And so there's a common contributor among all three of these. And so what we're seeing is that there is a cluster of matches where there's one common person between them all. And so then the question may be, now that we've started building this cluster, can we find more information in it? And here we now see this entire cluster of matches. So the database found 150 DNA matches within this one counterfeit drug cluster, where previously the laboratory found nothing. And you'll see that the data listed over there on the left ranged from November 2018 up to February 2020 and touches 21 different cases. And so there's one common contributor between all these different collections. So since we had that cluster of all those matches, the question is going to be, who is the common contributor there? So what we're showing here is from one sample, the vial and label edges, we were able to produce a allele profile. TrueAllele stores all the information, as I'll talk about in a second here, as probabilities, but for export to external databases like CODIS, it needs to be an allele profile. And so what we're seeing here on the right is an example of an allele profile. And what you're noticing is that it's a very strong profile. There's normally only one or two alleles at each location. As I'll talk about, CODIS uses an expected database match statistic called MME, and the MME for this allele profile would be 10 billion, 10 to the 10th, which would allow upload to even the national database. And in TrueAllele, where we're preserving all the information, we would expect a match statistic of 10 to the 30th to the true perpetrator. That was just one profile from one cluster of matches. Looking across all 103 counterfeit packages, we were able to find more information than just that. Across all the packages, we found 723 DNA connections. These are match statistics. We were also able to produce 6 CODIS profiles like the one I just showed you, and the actual interpretation time to do this was only a week. And this compares to the previous allele list method where they didn't produce information, they didn't get CODIS profiles, and that was while the data was being generated over a year and a half. This raises the question of why the previous analysis didn't produce much information. And it's not for lack of effort. It has to do with how the data is being represented and how much information is either preserved or discarded. And it has to do with a probabilistic database versus an allele database. An allele database has limits. It's limited in its upload and it's limited in its information, whereas a probabilistic database is much more specific, and so everything can be uploaded and information is preserved. And this has to do with how the information is stored. So on the bottom of the slide here, on the left, you'll see a list of alleles. On the right, you'll see a list of allele pairs with associated probabilities. And so this is the difference between alleles and a probability genotype. And the issue here is that if you look on the left, you'll see the alleles are 10, 11, and 12. And on the right, it's a 10-11 with 90% probability and a 10-12 with 10% probability. So, for example, if someone was a homozygote 12-12, they would be included in that list of alleles on the left, but it would not be included in the probability on the right. And so that's just a quick example of how the probabilities on the right are much more specific. They're not including like a homozygote 12-12 or a homozygote 11-11. There's more information in that probability. And so this also then in turn relates to what can be uploaded to the database. Because of the need for these limits on upload, the allele database uses something different than a probabilistic database. So in an allele database, there's this idea of MME, or moderate match estimate, and it's used more like a threshold limiting what can go up. You have to hit a certain level, like 1 million or 10 million, before you're allowed to upload to a allele database, because with there not being as much information in an allele list, if you upload everything, you're going to get way too many CODIS hits. You're going to get a lot of adventitious hits to whatever you're searching for. In a probabilistic database, there's this concept of KL, which is Kullback-Leibler statistic. And this provides the expected log likelihood ratio information, but is not a limit on upload. Everything goes up to probabilistic database. KL is just giving you a concept as you're uploading, how much information there is in the data before you even have a match. This concept of MME versus KL in information prediction has been previously presented on in 2019 at AAFS. And so what we're seeing in this graph is along the horizontal, we have 33 different hits from a database. And then along the vertical, we have log information, either a log likelihood, a KL, or a log of MME. And so these are 33 hits. These did not make it up to NDIS. These are all hits that came back from either SDIS or LDIS, state or local, where there was actually a match association found in TrueAllele. And so in this study, what you're seeing is that those blue bars that are kind of near the bottom are the log of the MME, and they're all somewhere around that level of 1 million. Then we have the KL from probabilistic TrueAllele database in orange, and then when the actual match was made, that's the green bar of log LR. And so what you're seeing here is that the orange and the green are much closer to each other. The KL is tracking and predicting the information from the match statistic. However, log MMA is pretty much always the same. And so this is just showing that for information prediction, KL is much more important. And so then once you have all this information on the database, what can you do with it? So I'm briefly going to speak about a couple other ways that you can use the database as an investigative tool. In addition to looking at counterfeit drug packaging, you could also look at illicit drugs, fentanyl, meth, with the idea being that can we use this as an investigative tool to find the original producers and suppliers. So the data here would be things collected from drug distribution sites, baggies, vials, scales, all the drug paraphernalia, and then the task would be the same. Take those, produce data from them, and then process it in TrueAllele, and then TrueAllele will automatically make all the comparisons and provide you with connections. And then those connections can lead to shared contributors, and you can produce profiles for database searches and try and get back to the source of these drugs. Then the same thing can be done for organized retail crime. Again, you're using it as an investigative tool where the question is, if there's a lot of things being stolen from retail stores and these items are collected, is there a shared contributor? Is there a central group doing this, stealing the items, fencing them, selling them elsewhere? And can we use the database to identify and link these criminals to the evidence and to multiple crime scenes? And so what you're doing effectively is building a map of the crime scenes that's connecting this person stole from these different stores and that these items have common contributors on them. And so it's giving you information and directing you where to look as you're looking into these large organized crimes. In conclusion, we were able to interpret in 1 week a year and a half's worth of data. We found 723 DNA matches and created 6 profiles that would be eligible for upload to CODIS. This didn't require extensive manual data review. Someone just quickly looked at the data, assigned a number of contributors, and from there on, everything is automated. The TrueAllele system automatically went, inferred genotypes, compared them, and then stored all that match information on the database for us to come in and retrieve. And so then we're just coming in and looking at that match information and finding the connections and exploring those connections. And this automated informative DNA database can catch counterfeiters and save lives. And then once you have this in place, it can be extended to other big DNA data problems such as illicit drug production and organized retail theft. Thank you for listening to our presentation today.